So good morning, good afternoon, good evening, wherever you're joining us from around the world, or some of our colleagues might like to say, bonjour, buenos tardes, and guten tag. It's great to have you here. I'm Jason White, CEO of LEC Partners, and I'm so glad you've been able to join us for our October Bioeconomy Webinar. Our goal for the webinar is to bring together leaders and innovators from across the bioeconomy, share practical and forward-looking insights, helping our team and our partners connect sustainability with business success. Whether you're a returning member of our community or joining us for the first time, welcome. We're happy to have you here. And JD, you can flip it over to the agenda. And here's what we'll cover today. A quick introduction of LEC Partners. I promise many of you heard this before. I'll keep it brief so we can get to the exciting parts of our meeting and cover how we help our clients succeed across the global bioeconomy. We'll have a celebration of two major milestones, at least major to us, our 30th anniversary that we're celebrating this month, and a recognition of 12 LEC experts nomination to this year's Bioeconomy 500 Leaders. We'll have presentations from our guest speakers, Dr. Harrison Yoon and Dr. Frédéric Kendirgi from Kula Bio. And finally, we'll welcome a few new experts that are joining our community from around the world, from Canada and Ghana to the UK and the US, expanding our reach and expertise. You know, personally, one of my favorite things is watching our community grow, especially across continents. It really shows how global and collaborative this field has become. Now, a little bit about LEC Partners. At LEC Partners, our mission is to help businesses navigate the bioeconomy, aligning sustainability with profitability. For more than 30 years, we've guided clients through technical, financial, regulatory challenges in biofuels, bio-based materials and chemicals, feedstocks, and helping scale industrial biotechnologies. From our headquarters in Cambridge, Massachusetts, we built a community of over 150 experts across 20 plus countries. 97% have advanced degrees, and we've evaluated more than 5 billion in bioeconomy projects. And what I think really makes us different is our breadth of experience, but really the depth of experience as well, and what we can bring to bear to help our clients. Our average consultant has 30 plus years of experience. So for us, we really believe we've built the most senior experienced team in the bioeconomy, and it's a real source of pride and differentiation for us. Many of you are joining us today as part of that community. And as part of that community, I'd like to celebrate a big milestone for us and recognize all the folks, LEC, whether it's our clients, our partners, or our experts that have joined us today. This year marks, or this month, October 3rd, marks 30 years of LEC Partners, and it's a milestone we're really proud of. Over the past 3 decades, our firm has grown from a small group of experts into one of the most trusted and experienced consulting teams in the bioeconomy, working on projects across 5 continents in every corner of the sector. Our growth has been driven by collaboration between our experts, our clients, our partners, like those of us joining us today, including the guest speakers, our great guest speakers today, who we've had the privilege to work with. So from all of us at LEC, thank you for being part of this journey. It's really the people that make this work, and is looking forward to the next 30 years. Speaking of part of this journey, many of these faces on this slide have been absolutely critical in growing and been with us for many years. But we'd like to take a minute to recognize that we're incredibly proud that 12 of our LEC experts were named among the Bioeconomy 500 Leaders for 2025. It's a recognition of individuals shaping the bioeconomy through innovation, investment, and leadership. And it's an honor that reflects the depth, talent, and impact of our team. And a big congratulations to those recognized this year, You continue to raise the bar across the bioeconomy. And, on a personal note, many of the folks, I won't, it almost doesn't have time to go name by name, but you know who you are. They've been with us along the way, and it really built the building blocks for what LEC Partners has become. And I want to thank you. So congratulations to everybody that was recognized for this achievement. Now let's get to the heart of our meeting. We're going to have a Q&A after our speakers conclude. So please enter your questions in the chat box. I will call on you, and we'll have an open mic. You get to ask your questions directly. I will note when we're asking, we do like to have conversations here, but also keep in mind the number of follow-ups or how long you take with each of the speaker. We usually have more questions than we can get to. So appreciate it. Again, put your questions in the chat box, and I'll call on you to ask your question. A little bit about our great guest speakers today. We're especially excited to welcome two leaders who've not only advanced the bioeconomy, but have also worked with LEC Partners as clients on real-world problems. First, Dr. Harrison Yoon, President and CEO of Kula Bio. Dr. Yoon has more than 25 years of leadership in biotechnology and agricultural biologicals. He's held executive roles managing global production and supply chains, and now leads Kula Bio strategy and execution, advancing sustainable agriculture worldwide. Joining him is Dr. Frédéric Kendirgi, Chief Technology Officer at Kula Bio. Dr. Kendirgi brings over 2 decades of experience in microbiology, fermentation, and cell biology. He leads Kula Bio's technology development, driving innovation in microbial cultivation and scalable biological systems for sustainable agriculture. We're thrilled to have both of you. I want to personally thank you for joining us. I'm especially excited about the innovations that you're bringing to agriculture. And with that, I will turn it over to you.
Thank you very much, Jason. It was a great introduction. I'm Harrison Yoon, as Jason introduced. I'm very honored to be part of today's LEC webinar. Today, we'd like to share how Kula Bio is turning scientific innovation into commercial reality through building a scalable, market-ready biological platform. Our presentation will have 2 parts. I will begin with a high-level strategic overview, focusing on the challenges, our approach, and how we are positioning Kula Bio for market adoption. Then my colleague, Dr. Frédéric Kendirgi, our Chief Technology Officer, will provide a deeper look into the underlying science and technology and how those innovations are driving measurable impact in real-world agriculture. Together, we hope to give you both the strategic vision and the technical foundation of what makes Kula Bio differentiated. Let's begin with the broader global context. And in this slide, most of you know this landscape very well, so I will keep it brief. Agriculture today faces a set of environmental and logistical pressures that the conventional chemical model can no longer sustain. Producing synthetic fertilizers is extremely carbon intensive. Each ton of ammonia emits about 2.5 tons of carbon dioxide to the air, and agriculture accounts for nearly 80% of nitrous oxide emissions worldwide. On top of that, the process consumes enormous amount of the fossil energy and contributes to soil and water degradation through runoff. The criticality of these challenges can be viewed differently depending on your perspectives, especially recently. Whether you focus on climate impact, energy security, long-term soil health, water contamination, political stance, or something else. But it remains true that our current fertilizer system is unsustainable in its present form and urgently needs more efficient and lower impact alternatives. Then we have external shocks, trade disputes, regional wars, political instability, all of which ripple through fertilizer supply chains, driving volatility in both price and availability. When you put it together, soil deterioration, growing demand, and unstable logistics create a real risk to global food security and long-term human prosperity. Next slide, please. Agricultural biologicals represent a key part of the solution as a sustainable and renewable way to nourish crops. But the pace of adoption has been slower than many hoped. Part of that comes from the larger economic environment we are all operating in. Rising tariffs have disrupted what used to be highly efficient global supply chains, and as a result, production and logistics costs have risen across the board. In today's agricultural market, the priority has shifted. Farmers and distributors are focused on securing reliable supply at fair and affordable prices. And in this context, the premium pricing structure that used to be accepted for sustainability or climate-friendly product is becoming harder and harder to justify. Beyond economics, there are still scientific and practical challenges. We don't yet fully understand how microbes interact with the plants or with each other under real-world field conditions. And with the regulatory uncertainty, especially when it comes to gene-edited or genetically modified strains, companies and growers alike tend to make a cautious approach. Manufacturing adds another layer of difficulty. Scaling up living organisms is very different from scaling up chemistry. It comes with risks like contamination, genetic drift during seed train expansion, and the constant balancing act between cost efficiency and consistent product quality. And even after production, agricultural biologicals face real logistical constraints, short shelf life, temperature sensitivity, and distribution delays that can cause the products to miss a farmer's planting window. These are exactly the kinds of challenges that Kula Bio was created to solve, bridging the gap between the sustainability and commercial practicality to make biological solutions that truly work at scale. Next slide, please. Kula Bio was spun out of Harvard in 2018 and is based in Natick, Massachusetts, in the greater Boston area. Our mission is to transform agriculture and advance food security through sustainable innovation. We do that by combining microbial excellence with manufacturing innovation that makes those microbes commercially scalable. In late 2024, we launched our first two products, Kula N, our organic formulation, and Kula Next, our conventional version. Both products are already performing strongly in the field. In late 2025 this year, we're going to add wettable powder version, a kind of dry formulation products to improve shelf life, simplify logistics, and open global distribution channels. Next slide, please. Our operations today already demonstrate both scale and impact. From a 420 square meter, roughly, which is 4,500 square feet, small manufacturing facility, we produce more than 100,000 liters of microbial culture annually, achieving 99.939% microbial purity at minimum. This is a level of consistency that's rare in agricultural biological manufacturing. The output translates into roughly 17 tons of dry product, enough to replace about 70 million pounds of nitrogen fertilizer every year. On the farm, its treated acre reduces around 120 kilograms, which is 265 pounds of carbon dioxide emissions simply by replacing synthetic nitrogen. Since 2023, we have completed over 200 field trials across multiple crops and geographies. At full utilization, our capacity can support more than 8,000 square kilometers, which is roughly 800,000 hectares of farmland with 40 pounds of nitrogen per acre replacement. So this is not just a proof of concept. It's a functioning, repeatable system with measurable environmental and economic value. Next slide, please. Our advantage comes from two reinforcing pillars. One is microbial excellence, and the other one is process innovation. First, our proprietary strain, Xanthobacter autotropicus, stands apart from typical nitrogen-fixing bacteria. Most nitrogen fixers depend on carbon compounds released by plants. We call them root exudates. Plants generate these compounds through photosynthesis. They use a portion for their own metabolism and send the rest to the root zones or rhizosphere and release them to attract the beneficial microbes from the soil, which is called the rhizodeposition. The root zone is a very fascinating microenvironment. It's where an incredible amount of the biological trade and interaction takes place between plants, microbes, and the surrounding soils. What makes our strain Xanthobacter autotropicus different is its metabolic versatility. It can use those plant-derived carbon sources or organic carbon in the soil, just like any other microbes, but it's also capable of switching to a complete different mode of energy generation when the conditions become harsh. So in nutrient-poor or degraded soils, it activates its hemolytic autotropic pathway, allowing it to draw energy from inorganic sources like a hydrogen ion from inorganic materials in the soil and the carbon dioxide from the air and store them intracellularly as a reserved energy source. In simpler terms, this means it does not rely solely on the plant for survival. It can power itself even when organic carbon is limited. That makes it especially valuable in marginal, sandy, or semi-arid soils where traditional fertilizers and most microbes struggle. You can think of it as a living soil factory that keeps producing steady, on-demand nutrition right at the right zone, the root zones, even under challenging environmental conditions. That's the microbial excellence part. Second part is our MSHD platform, which stands for minimum scale, high density. It fundamentally redefines how agricultural biologicals, especially microbial products, are manufactured. Traditional fermentation depends on large, centralized tanks that require significant upfront capital investment. The conventional idea of scale-up is straightforward. Increase the vessel size to increase the production and lower production cost. That model works very well in pharmaceuticals, where I spent much of my career as a bioprocess engineer, focusing on process design, process optimization, and scale-up execution. In pharma and nutraceutical manufacturing, high cost can be justified because the product value is also high. However, in agriculture, that's a different world. That equation doesn't hold. Cost efficiency is absolutely critical, and when traditional scale-up methods are applied, manufacturers often face a tough choice between keeping costs down or maintaining quality and consistency. Unfortunately, very much often, quality is the part that gets compromised. My colleague, Frédéric, who also came from pharma and vaccine industry, and he and I both saw that this traditional scale-up model could never make agricultural biological manufacturing economically feasible for agriculture. We needed a completely different approach, which is to deliver high performance and low cost at the same time. That mindset led to the creation of the MSHD platform. When we examined the agricultural biomanufacturing process from end to end, it was clear that several parts of the system had to be re-engineered to meaningfully reduce the cost of goods. The key was to break away from the idea that scale only comes from increasing volume. The MSHD platform optimized both volume and cell density. It enables us dramatically higher productivity within smaller, more efficient, compact bioreactors. In other words, we scale output, not infrastructure. We have designed a modular system that achieves ultra-high cell density in compact bioreactors. This allows us to deploy smaller, decentralized production units closer to demand and end market, scale them out quickly, and maintain consistent precision-grade quality consistently above 99.99% microbial purity as our internal in-house QC criteria. This approach transforms agricultural microbial manufacturing into a flexible, capital-efficient model that fits the realities of agriculture, where cost, quality, and scalability must coexist. Together, the strain and the process create a platform that's both scientifically advanced and commercially viable. Eventually, it's going to be one that competes directly with the chemical fertilizers in cost, consistency, and scalability. Next slide, please. So where does all this innovation lead in practice? We think about the application in two horizons. The immediate opportunity to make existing farmland more productive, and the long-term goal for opening up new land for sustainable cultivation. In the near term, we focus on sustainable intensification. This is for helping farmers get more value from land they already manage. Our first approach is what we call sustainable replacement, substituting part of the synthetic nitrogen with our biological solution while maintaining yield and reducing environmental impact. This is particularly effective in soils whose fertility was declined after years of heavy fertilizer use. Our second approach focuses on performance enhancement through what we call a boosting effect. So by improving nutrient uptake and stimulating root activity, our microbes help crops grow faster, stronger, and more efficiently, translating directly into higher yields and better returns for growers. In this model, return on investment becomes the key measure of success, as the value is realized through both productivity gains and in input efficiency. Looking further ahead, the real transformation lies in agricultural extensification, expanding food production into underused or degraded land. Because our microbe is a cumulative autotroph, it can power itself in nutrient-poor, sandy, or semi-arid soils where chemical fertilizers often wash away or underperform. So this allows farmers to reclaim unproductive areas, expand the cultivation without deforestation, and build the resilience against the climate stress. In short, our technology supports today's farmers in producing more with less, while also unlocking tomorrow's farmland potential. So to summarize, Kula Bio bridges a long-standing gap in agriculture, which is bridging together biological precision and engineering discipline and economic scalability. We are proving that sustainability and profitability don't have to compete. They can reinforce each other. So with that, I will hand things over to my colleague, Dr. Frédéric Kendirgi, Kula Bio CTO. He will walk you through the deeper science behind Xanthobacter tetraficus, our proprietary strain, and explain how our MSHD manufacturing platforms turns that science into consistent high-performance products. Frédéric, over to you.
Thank you, Harrison. So I just wanted to introduce you all to this kind of newcomer in the ag biological field, which is Xanthobacter autotrophicus. It has been studied for quite a while, and its capability of capturing nitrogen from the air has been well documented in the scientific literature. The question that my team wanted to answer when we chose this particular microbe to move forward with was whether or not the nitrogen that it was able to fix from the atmosphere was actually the actual nitrogen that made it to the plant and was not used for its own benefit or benefit of other commensal microbes in the environment. So next slide. So quickly to summarize this particular microbe, as Harrison mentioned, it is a chemolethal autotroph microbe. It has the ability to capture carbon dioxide from the air and use that as a source of carbon. As I said, its genome has been well characterized. It has all of the machinery to fix nitrogen. It is associated with the rhizosphere. You can see in the bottom left a fluorescent microscopy image where the root is colored in blue and Xanthobacter autotrophicus is in green. You can see it coating the root tips of the plants. It is free living at this point. We do not have any evidence that it is an endophytic bacterium. And it has, as we mentioned, agricultural benefits. This was shown in a trial and very early on when we started this endeavor. That's the middle panel there, where we looked at lettuce in pots, and we cultivated this lettuce with the presence of a standard amount of nitrogen. We looked at the above ground biomass once we harvested those particular plants. And what we also did was to remove about 80% of the synthetic fertilizer from the fertilizing regimen. And what we saw is a reduction in the above ground biomass by roughly 80%, which made sense. However, when we added the Xanthobacter to the equation, you can see in green how it rescued the deficiency in above ground biomass, meaning that at least we could replace 20% of the synthetic fertilizer application by applying this particular microbe. Now, as I mentioned, it is a well-known nitrogen fixer. It is a non-spore-forming microbe, so its way of surviving in environment, as Harrison mentioned, is to produce a intracellular biopolymer called PHB, polyhydroxybutyrate. That particular polymer, and we have evidence to show this, is key in driving the nitrogen fixation machinery. Nitrogen fixation by nature is not something a microbe wants to do if it has other opportunities to get nitrogen from another source. It is very energetically taxing, and it has to have an immediate benefit to the microbe in order for it to do so. In this instance, by using a non-genetic engineered microbe, this is what we have, it's the wild type strain. We've developed a way during our fermentation process to supercharge it with this particular biopolymer. And that allows it to drive and fuel the nitrogen fixation machinery without really it without needing any kind of other alteration to the life cycle of the microbe. Now, as I said, we know that it fixes nitrogen. The literature tells us that it fixes nitrogen. It presumably secretes it as an ammonia form. The question is that N in that molecule of ammonium actually coming from the air, or is it maybe taking some of the fertilizer and transferring it to the plant? So what we decided to do is an experiment using a stable isotope, N15, in these mason jar type of contraptions, where we grew lettuce plants in soil, and we either added Xanthobacter or not in an environment that was deficient in synthetic fertilizer. So on the what you see in the last graph on your right is the results of that experiment. We looked at the abundance of N15, background abundance. It's a natural isotope. We then looked also at the abundance of this natural isotope when we cut the amount of fertilizer, roughly around 75% in the purple bar. And then when we added the actual, to that 75% grower standard practice, we added Xanthobacter to the mix. You can see that in the leaves, and these were all as determined in the leaves of the plants, a significant increase in N15. That told us that the gaseous nitrogen that was present in the headspace of those jars made it through to the plant via our Xanthobacter. So it wasn't really that Xanthobacter was taking on some of the fertilizer nitrogen and passing it on to the plant while fueling its own needs in nitrogen fixation. It was truly taking what was from the air and passing it on to the plant. That was a quintessential proof that the microbe transfers gaseous nitrogen to the plant. Now, the question was, where does it do this? At the end of the day, we know that it is root associated. The microscopic image that I'm showing here shows it associated with the rhizosphere. But does it really fix nitrogen there, or does it just fix nitrogen anywhere all the time and just eventually the plant will find that particular nitrogen for it to uptake? So my team, if you go to the next slide, please, devised a lab version of this strain where we actually coupled the green fluorescent protein to one of the promoters that is essential in triggering the nitrogen fixation genes. So that meant that when nitrogen fixation occurs, this protein would be expressed and therefore fluoresce. So our bug will basically fluoresce during nitrogen fixation, and we have evidence that it does so. And when there is excess amount of nitrogen in the medium, for example, you see that that gene being repressed, so there's no glowing and no production of GFP. But as soon as we reduce the amount of nitrogen in the growth medium of the microbe, it suddenly starts kicking off that nitrogen fixation machinery in order to produce its own needs, or for nitrogen, its own needs in nitrogen. So we then took that culture and we applied it to lettuce plants. Here you have on the left-hand side a root of a lettuce plant. The orange goo that you see is actually the culture itself, the microbes themselves. It's a fairly slimy culture that coats the roots when you do that in vitro type of assays. And you can see there's some fuzziness around that root. Those are the root hairs. So when we switched over to fluorescent microscopy, you can suddenly see that all of these root hairs are actually glowing green. That meant that nitrogen fixation, catalyzed by our microbe, was actually occurring at the root hairs. This is important because it tells us that this is actually the way the microbe is able to transfer that atmospheric nitrogen that I mentioned earlier into the plant through the root hairs. So light nitrogen fixation occurs in situ upon association with the plant. Next slide. So Now, this is fine. This is a nice lab model, but at the end of the day, can we actually produce enough of this microbe to be able to make this particular endeavor commercially viable and feasible? Next slide. So standard manufacturing of ag biological through fermentation has been going on for a very long time. The process uses multi-stages bioreactor that go from a small scale to maybe tenfold increase, so maybe a 1 liter to a 10 liter to 100 liter, so on and so forth. Not necessarily needing to be a tenfold increase, but there is what we refer to as a seed train to actually exponentially increase the volume of culture of product to be sold. Now, in these particular bioreactors, you can see in red, the product volume increases as the scale of your reactor increases, of course, that's the intent. But the cell density in each of these reactors pretty much stays the same. You just grow it to a certain level, for example, maybe 1 billion cells, and then you bring it back down, you use that as an inoculum to the next reactor, and so on and so forth. So it's a bit of a seesaw. This is a traditional way of producing or increasing your scale of production. It has some serious disadvantages to it. It is simple. That's one of the advantages. It's a very well known, very well characterized process. So nothing wrong with that. The challenge, though, is that it does require larger and larger facilities. We're talking perhaps at the end of the day, depending on how extensive the company wants to be, hundreds of thousands of liters of scaled bioreactors. So, and that requires basically larger and larger facilities. At the same time, from a process point of view, as you basically subculture this particular product to increase your scale, you run the risk of introducing contamination, genetic drift as you keep on multiplying the microbe throughout the process. There are a lot of places when things go wrong and having you needing to go back to square one and start the process over again. What we wanted to do at Kula Bio is take some inspiration from the pharmaceutical industry, as well actually from the biofuel industry, and apply that to a microbial product. So in our manufacturing process, which is what you have on the right-hand side, we actually start and finish with only one reactor. And a fairly, less than about 1,000 liter scale bioreactor. What you do is that you're able to inoculate this bioreactor with your seed culture, and you grow it up in that bioreactor in a way where you can achieve fairly high level of cell concentration, up to 1 trillion cells per ml. By allowing us to increase the cell density, we then work to limit the liquid volume of the particular product, which at the end of the day is applied, diluted into the field. So it's not like you're changing the amount of product applied to a plant. You're basically producing a concentrate that will allow you to not only save on storage cost, but on shipping cost as well. And by having only one scale bioreactor, you are working in a closed system as much as possible. You have better control over contamination issues, as well as over any kind of genetic drift that could occur during the process. Next slide. So how does that actually work itself out when we're talking about parity with current urea prices? At the end of the day, our mission is not to produce a product that is more expensive for the farmer to use. What we're trying to achieve is parity with current nitrogen prices. So on the left-hand side, you actually have the traditional manufacturing process, which we talked about, where in order to achieve as parity with the urea prices, for example, you need to increase volume. In our particular instance, is what you have on the right-hand side, rather than increasing the volume, you increase the cell density. You make the product much more potent volume per volume compared to the traditional method. And this is how you end up getting closer and closer to parity with the synthetic fertilizers. Next slide. So this is all nice. It's, we've demonstrated in lab scale that it works in plants. We actually have a manufacturing facility, so we are able to achieve those very high cell density in fairly small scale bioreactor for the industry, of course. But does it actually work in the field? So what we've done is we've actually applied the product in two ways. Next slide. One way was as a replacement of about 20% of grower standard practice, 80% GSP, in different crops and different geographies in California and Florida, and as well as within those states, different plots or lands. We've looked at lettuce initially. We were focused on high-value crops, lettuce, tomato, peppers, and berries as well. Here I'm showing you results that we've gotten for those three particular crops by replacing up to 20% of current grower standard practice. We looked at marketable yield. Initially, our intent was for the farmer not to see a difference when they replace their 20% synthetic fertilizer with our product. However, we were happily surprised to see that in some instances, we were actually getting better marketable yield of these particular products than the standard 100%. That was not the intent. This is why you see that there's a bit of variability from, plus 2% to all the way up to 10.3%, depending on the crop. This is very interesting. That was not, as I said, the intent initially, but that meant that you bring a biological to the soil. There are other things other than nitrogen fixation that biological brings. We all know the concept of probiotics. That probably has something to play in here. Now, the interesting aspect of this is when we tried the other version, which is trying to add on top of the synthetic fertilizer, Kula end products, which contain our particular product. Now, you have to remember farmers are not always very eager to change their practice. Our product intercalates itself in their fertigation process. They don't need to do anything different. But coming to a farmer and asking him or her to actually go and forgo 20% of their current fertilizer regimen is not something that they're very eager to do on day one. So we also tried to penetrate the market using the boosting aspect, next slide, where we've added Kula N on top of that 100% GSP to the same amount that you would add for the 20% replacement. And in these cases, what we saw, again, in different geographies, we saw that you actually did have a increase in marketable yield, quite significant. Now, again, to be transparent, it doesn't always work as expected. You see there, for example, in Patterson, California, for tomatoes, we only had a 2.5% increase. But in other geographies and other areas, we did get above 10%, quite a bit. Now, it's going to depend on crop. It's going to depend on farmers' practice. It's always going to depend on the climate, the stress. So one has to expect variability. But we're happy to see that throughout those particular crops, we actually had a significant boosting effect, which kind of convinced the farmer a little bit more to adopt the product and hopefully eventually try and use it as a replacement rather than a boost. Now, I want to point out something that is quite interesting, especially is the result that we got with celery. In that particular field, celery was grown in a fairly sandy soil, a very leach-prone soil, doesn't retain a lot of nutrients. And we got a very, very good outcome. So that prompted us to think about the use of our product in environments where you actually have poor retention or low amounts of micronutrients. Remember, this microbe is fundamentally able to survive in the environment by itself. As Dr. Yoon mentioned, it can actually fix CO2 and use carbon from CO2. It can use hydrogen from the soil, and it has basically all the ability to grow without really needing a very rich environment. So we decided to test this product in a desert-like environment I think we have a few already.
Yeah, we have quite a few. Thank you so much. I appreciate you sharing with us, the changes in in innovations in agricultural and what are some of the most important issues really in the world as far as food security, sustainability. And Harrison, you said it right, and it's one of the ways we think about things is connecting sustainability with profitability. And that's really one of the major challenges that I think all of us in the industry face. So I appreciate it, gentlemen. Very interesting. Kind of right up the alley of what we focus on is really important to us around here, making those connections. So I'll go ahead and open it up. We have Mike Stickland Mike.
Yeah.
If you
hi.
could, you have several questions. If you could
Too
rattle
many, yeah.
No, I mean,
Hey,
I think
guys.
it's great. It's great. But yeah, go ahead, Mike.
No, Harrison, Frederick, this is exciting. I'm really excited to see this. I live right down the street from Contever Agri-Science Research Center, and everybody's working on nitrogen fixation with alternates to fertilizer. So
Yes.
I got too many questions to really go into, but are you working I see you're doing corn in China. Are you working on other row crops like soybean and corn and soy here in the U.S.?
Yes, we are. Potato and corn are go-to at this point in row crops in the U.S. Yes, we're definitely looking. It's not just in China. Absolutely.
Fantastic. Okay. And then do you see that obviously this is a way to reduce carbon intensity of the corn. We do a lot of work in biofuels. So as a route to reduce the carbon intensity of the feedstock, this is really exciting. So look forward to seeing more to come. Thank you.
Absolutely.
Thank you very much.
Great. Thanks, Mike. Well, I believe we have Greg Potter. You had a question.
Greg had to jump to another call, but we can ask his question. Yes.
Well, Greg, I'll read Greg's question. Maybe you can see it, but it says your field trials highlight in the presentation deck look to be warmer climate zones, Florida and California. Is there any chance that cooler climates may negatively impact product efficiency? How does your product shelf life compare to chemical fertilizers?
So it is a microbial. So any microbe, as the temperature goes down, will obviously slow its metabolism. However, we have done trials also in the northern states in the U.S., and we haven't had any issues during the normal growing cycle. We've also done trials in the U.K. They seem to be doing well over there as well. So I'm not, as long as agriculture, as long as the plants are happy and actively growing, this microbe will follow suit. I'm not worried about that at this stage. Of course, if you're talking about freezing soils, that's going to be another issue.
Yeah.
The reason why we have run some focus on Florida and California and we have focused on specialty crops and organic market. And the reason is actually the fertigation system and also the irrigation system is more favorable to microbial activities, which is actually drip irrigation. So close to the root zones through the drip irrigation is the best way to add the small amount of the microbial product closer to root zones to see the efficacy. And also from a strategic perspective, the row crop field, from our perspective initially, rather than the blue ocean, but red oceans, because all the kind of biological companies are trying to rush to the Midwest for focusing on corn, soybean, and wheat. I know there's huge acreages, but we wanted to obtain good success story from relatively smaller business field, which is specialty crops. And then we're going to try to start from there to expand our business scope to the row crops. That's why, as Frederick mentioned, we have been running some trials in Midwest with the corn and soybean and wheat as well. We are collecting more data to gain the higher confidence in our product efficacy from the row crops as well.
Yeah, that makes a lot of sense. We have Maya. Are you still with us? I guess I'll read Maya's question as well. She says, soils and crops are often drenched with pesticides. How have your microbes withstood the more common pesticides and still be effective?
No problem whatsoever, actually. At the level, of course, concentrated pesticides cause any would have any issue, but at the application rates, we haven't seen any major problems. This is why we asked the farmer to actually naturally intercalate our product in their fertigation process as well as in their biocontrol process without any significant issue. It is a fairly robust microbe.
Okay. And we have a question from David Peterson. David, would you like to ask your question?
Absolutely. Thank you for putting together this presentation and all the interesting work. I actually have the question I posted, and I have a follow-up question due to some of my experience in developing some agricultural products as well. But I'm sort of intrigued by the limitation on the chemical fertilizer replacement number that you have bumped against. Is that something that's sort of arbitrary, or what can you do to increase that number to a higher replacement percentage?
So we've had instances where we could go much higher than just 20%. I gave you the 20% because that seems to be, across the board, a pretty standard, decent response from the crops. So we've had instances where you can go 30%, if not 40% as well. But those are kind of peppered here and there. Now, if you were to ask me, how would you want to do this? So my R&D team is actually working on finding ways to beef up the nitrogen fixation capability of this microbe without doing any kind of genetic engineering. And we have ideas on how to do that. And so that will translate into probably greater replacement in the soil. Now, if you want to go hardcore, I would say genetic engineering, but this is not what I would like to do in order to beef up the nitrogen fixation capability.
Sure, that makes sense. It also, from the experience I had before, especially when you're looking at specialty crops like this, fungal attack, different types of fungi can greatly undermine the growth of crops.
Yes.
Have you found that this actually helps to make the crops more hardy or more resistant to fungal attack?
So we don't make any biocontrol claim in any way, shape, or form regarding this product. However, you have to remember that a healthy plant will rebound much faster from those types of stresses than an unhealthy plant. So it's not just biological stress, but it could be abiotic stress as well. So either flooding or drought. A lot of the farmers come back to us and say, hey, you should say that about our product. That is not a claim that we make, but it is a natural observation. When you have a healthier plant, it will actually rebound better from any type of stress.
True. Good work. Thank you.
Thank you.
Thanks, David. And I think we have time for one more question. Kevin Hadenbach, would you like to ask your question?
Yes, hi. Harrison, Frederick, really nice presentation. I had a question along the lines of, you mentioned you use a single bioreactor, say at 1,000 liters. And did you estimate the cost of goods versus a traditional fermentation process with the seed train and higher volumes? And what does that look like for the economics side?
Harrison, do you want to take on?
So our always benchmarking reference is a synthetic fertilizer cost of goods at market price, because we understand the urea's kind of price range depending on the fluctuation of supply chain up and down a lot. But in the U.S. market, roughly the 65 cents to the $1.25 per pound of nitrogen, and up and down all the time. But if you're going to go to some other foreign countries, for example, China, there's some provinces considering the subsidies from the Chinese government, sometimes at 20 cents per pound of nitrogen, the US dollars. And also the lowest one is 12 cents per pound of nitrogen. So that's why we wanted to spread our advanced technology and the product out of the technology across the entire world to make some contribution. And also this is great payback from us to the farmers. That's why we always try to maintain low production cost. Due to the MSHD's kind of efficacy, we already in a very good positions to have a really good pricing position in cost of goods, and to compete with synthetic fertilizer in any country in any pricing structure in different countries. That could be kind of our answer for you.
And let me let me add one more thing, because you were comparing traditional fermentation with the C train versus ours. At the end of the day, most of the cost, especially in the US, is associated with labor. Remember, if you're dealing with only one reactor compared to having an entire C train, you haven't. We have a handful of people can actually manage what normally a standard fermentation facility would need, 10 or 20 people. So you do have a significant savings there at the end of the day when it comes to COGS compared to traditional and the MSHD process.
Thanks, Frederic. Yeah, thanks for the feedback. And you have a liquid formulation such that you don't incur DSP costs as well?
Yes, and actually we considered all the part of different process from the upstream down to the downstream process. So considering that, I think that we are very superior positions to be flexible with the pricing strategy.
Great. Thank you, Harrison.
I think that's all the questions or time that we have. Thanks for your question, Kevin. Harrison, Frederic, really appreciate you taking the time to show us what you're doing over at Kula Bio to drive sustainable agriculture. And obviously, if we can ever help you, let us know. We're happy to work with you in the past. And I know this is really beneficial for our team. So I appreciate your time. We're going to introduce our new experts. We are absolutely welcome to hang on, but I imagine you probably have some things to do today. So. Thank you so much, gentlemen.
Thank you very much. Thanks a lot for having us. Thank you, audience.
All right. Now here's one of my favorite parts. Say this too frequently. Welcoming our new experts to the group. We have several new experts that are going to join us today, and each of everybody on the phone, everybody on the team plays a vital role in how we deliver expertise to our clients, strengthen our presence in the global bioeconomy. So I'd like to introduce a few new team members today, give them 2 or 3 minutes to talk about themselves. And first up, we have Viraj Patel. He's in Calgary, Canada, and has his focus areas are energy, hydrogen, biofuels, CCUS, and I'll let Viraj tell more about himself in more detail than I could give him credit for. So, Viraj, are you with us?
Viraj, please unmute your mic.
Are you there, Viraj? Is he on, Elena?
Yes, he's on.
Okay. We'll give it another few seconds, and then maybe we'll go to the next person, and then come back to Viraj. Sometimes, as Elena knows, sometimes I take a second to get off mute myself. All right, let's move on to Mike, and we'll loop back and see if Viraj can join us. We have Mike Van Houten from Illinois, and his focus areas are agricultural processing, carbon scoring, biofuels, operational optimization, and project management. And Mike, would you like to take 2 or 3 minutes to introduce yourself?
Sure. Good morning, everybody. Mike Van Houten, 48 years in running plants, optimization of plants. So flour milling, wet corn milling, masa milling, you have a lot of milling stuff, right? All ag processing into food. So lots of food experience. Ran an ethanol plant for 10 years. So I have good experience in the ethanol world. Last few years been working on business development, and that included carbon scoring of these ethanol plants and how we document that. Ran the project for carbon capture and sequestration. So that is in the after I retired, they're continuing. They're in the permit process. So as many of you know, that is another 2 to 3 years process to get through the EPA. So lots of experience in project management, maintenance and reliability, all the things that go with a plant and running a plant and keeping it safe and keeping it efficient. So glad to be on the team. Thanks, Jason.
Thank you, Mike, and you certainly you fit right in with our team as far as your deep experience and kind of practical experience, I think, as well. So I appreciate you taking the time to introduce yourself.
Yeah, I was thinking I'm moving the average up a little bit. So
we don't mind that, Mike.
all right. Thank you.
Thank you, sir. Next up, we have Lambert. I'm going to try to get this right. Ali Jinya. It's coming to us from Ghana. Israel. Its focus areas are climate change, natural resource management, agribusiness, cocoa certification, and carbon projects and biofuels. Lambert, are you on the call with us?
Yes, Jason. Can you hear me?
Yes. Thanks for joining.
Yes. Thanks for having me. I want to say congratulations to LEC Partners for 30 years of turning opportunities into sustainable results. I didn't know this. I just read about it, and I also came and met it on the webinar. So I'm Lambert Aliginia. You tried it. I think you did well with my name. Thank you. Climate change and sustainability specialist with a long term of about 10 years experience and cocoa certification and sustainability, supporting traceability system through cocoa supply chains. I've worked with carbon projects development, that is afforestation and agroforestry, taking up on-site assessment, and at a point in my career, led the development of
Lambert, you're breaking up on us a little bit.
Yep. So I happen to work with Ecom West Africa within the cocoa industry, cargo cocoa sourcing, and I work with GOL on the carbon projects development. I also implemented IACC EU Plus and CORSIA certification for the European markets, that is for biofuel crop JETROFA. Implementing end-to-end certification cycle for the biofuel crop in my period of working with GOL. And I'm currently working as an independent consultant, helping farmers and organizations across West Africa and Southern Africa, implementing biofuel projects and also supporting other clients in the on-site assessment of carbon projects. Briefly, I will say this is my experience, and I am also a founder of Art Beyond Limited, which is a young company supporting farmers and organizations in Ghana and West Africa within the cocoa industry, as well as the nature-based solution sector. Thank you.
Thank you, Lambert. Thanks for joining us, and I think it was a timely call to be joining us for the webinar topic. So appreciate it and welcome.
Thank you, Jason.
And next we have Aminu Owanani Koko. Sorry, Aminu, I messed up your name. Are you there to?
I'm there.
To introduce yourself.
Yeah, that's right. Thank you for having me. As you said, my name is Aminu Owanani Koko. So I'm based in the UK. Originally, I'm from Nigeria, and I've worked in the UK, US, Australia, and Nigeria as well. So my area of expertise has been listed here. Basically, what I'm going to add is my combination of academic expertise with entrepreneurship. So in the UK, I'm the owner and CEO of our resources emissions, where we are mapping emissions in the food supply chain. So I combine different expertise, including business management, and other areas have been listed, as I've said, such as biomass and agri-food waste handling, biofuel production, food engineering. I have my bachelor's degree in food engineering, and I've designed some equipment for food applications, such as meat mincer or meat grinder. And also I'm an expertise in life cycle assessment. So using it to uncover the emission spot before starting the actual decarbonization process. And also I have experience of different model. One of them is viscoelastic model. So using it to study the stress relaxation properties of biomass and waste materials, especially in agricultural sector. And when I was in the US, I worked for United States Department of Agriculture in collaboration with Kansas State University. So it's part of a research program. So I was a graduate research engineer when I was there. And as I've said, I'm from Nigeria, currently based in the UK, but I'm willing to travel around the world to do the project for LEC Partners. Thank you so much for having me.
All right, thank you so much for joining us, Aminu. I think we have, is Elena, I know Dr. Sarah Glavin was going to try to join us.
Yes, she's with us today. Yes, she has joined us.
Fantastic. Yes. I appreciate the going in, above and beyond to join us. Sarah, are you there?
I am. Can you hear me?
Absolutely.
All right. Well, good morning, everyone. Wow, this is a really, I'm so impressed by everyone's expertise. It's actually much broader than my own. Thanks for allowing me to be part of the group of experts, Jason. My expertise really falls more on the early research and development side of biomanufacturing, and I spent 16 years in government as a research scientist in the Department of Defense. And they became very interested in this topic over the last 5 to 10 years. So I have experience in program management, large government programs, government contracting, as well as doing the research myself. And then about 18 months ago, I joined the final year of the Biden-Harris administration as the top scientist for the bioeconomy. So I have some policy background as well. And right now I'm taking a year on a fellowship at Princeton in the Andlinger Center for Energy and the Environment. So this year, taking a lot of time, spending a lot of time thinking about the role of biomanufacturing and industrial decarbonization and carbon capture, and also just growing our United States manufacturing base for the bioeconomy. So yeah, happy to engage with anyone and just grateful to be part of this amazing group.
Great. Thank you so much for joining us, Sarah. Obviously, you know, your background is most of our experts very impressive, and you know, having some more help on the kind of the R&D end of the spectrum it certainly adds to our expertise here. So thank you so much for joining us. And I assume I'm going to call, Elena's probably politely telling me that Viraj is not available.
No, he wasn't able to
darn.
Unmute.
Maybe next time then. Anyway, welcome to all the experts that joined us. Certainly, if you're on the call and you want to connect, and I definitely encourage this, if you're part of the LEC Partners team and want to connect with some of the consultants that presented themselves today, LinkedIn's a great place to do it. If you want their contact information, we can reach out to Elena. We like to make connections here. It help it helps rise all boats, if you will, lack of a better way of saying that. So I'll close. First of all, thanks for everybody joining us, and I appreciate you holding on. We went a little bit longer, but I think the topic was extremely interesting. We had some great questions. Thanks for joining us. You know, your engagement and your participation in these webinars really is what makes it work. It's not just the speakers, but everybody joining us, engaged, asking questions. And I think we all, well, at least I'll speak for myself. I learn a lot in this process, not about our experts, about our clients, about the science, about what's going on out there. And I hope you all do too.